Hello everyone, welcome to today's webinar. I'm Manny Marone, Associate Editor at CNEN, and I'll be moderating today's event. This webinar is titled, Evaluating Extractable and Leachable Profiles of 3D Printed Medical Devices, and is being sponsored by Waters Corporation. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN Online after the live webcast. The presentation today is being sponsored by Waters Corporation. Waters is the premium brand in the analytical instruments industry, creates business advantages for laboratory-dependent organizations by delivering practical and sustainable scientific innovation to enable significant advances in healthcare delivery, environmental management, food safety, and water quality. Waters products are used by pharmaceutical, health sciences, biochemical, industrial, academic, and government organizations working in research and development, quality assurance, and other laboratory applications. Waters UPLC, HPLC, UPC2, and MS instruments and consumables are used in a broad range of industries to measure the chemical, physical, and biological composition of materials. And during the presentation, we'll be hearing from Dr. Joel Rindelab, who is a principal chemist at Pace Analytical Life Sciences in Oakdale, Minnesota, where he developed an extractable and leachable testing program within a GMP-compliant laboratory, focusing on support for the medical device and pharmaceutical industries. Joel has extensive experience with mass spectrometry, applications, and chemical characterization techniques, receiving a PhD in analytical chemistry from Purdue University. In addition to method development, scientific education, and journalism, Joel is also a successful researcher and continues to publish his works in peer-reviewed journals. I will now hand it over to Joel Rindelab.
Thanks for the introduction, Manny, and thanks to Chemical and Engineering News for putting on this webinar and Waters for sponsoring. And today I get to talk to you a little bit about some work that I did looking at extractable and leachable profiles of 3D printed medical device. So once again, the introduction. I work for Pace Analytical Life Sciences, which is a contract lab focusing on chemical and microbiology testing for the pharmaceutical and medical device industries. As Manny mentioned, I'm based in Oakdale, Minnesota, which is just outside of St. Paul. And we also have a lab in San German, Puerto Rico, and coming soon to Somerset, New Jersey. And we also do a lot of E&L studies here at Pace. And really the whole idea behind extractable and leachables is to investigate product safety. We'd like to ensure that only the intended application is occurring, as well as potentially harmful chemicals are not present in your product or are leaching out of them. So one extractable and leachable that has gotten a lot of media notoriety in the last decade or so is BPA, or bisphenol A. And the reason is that it mimics the actions of estradiol, which is the main component of estrogen. So when considering extractable and leachables, it's important to think about not only the application of the product, but the patient population as well. So for instance, if I were to drink a water bottle with BPA plasticized polycarbonate, for instance, there isn't really a large probability that BPA is going to leach out into that water at room temperature. On top of that, I am a grown adult, or at least physically, so this slight hormone imbalance really isn't going to have a large effect on my day-to-day life. However, if we are to look at a newborn child, for instance, And we are going to use a BPA plasticized baby bottle to deliver milk to the child. And this would be milk that has a high fat content. And let's say it's put in the microwave. We have then greatly increased our chances of BPA leaching into that milk. And on top of that, a small child is greatly dependent on the hormone balance for development. So therefore, the introduction of BPA into their system could alter their development as a growing child. So as you can see, extractable leachable studies are very important considerations when making an assessment on product safety. And extractables and leachables can come from really any part of your product development, whether it's inherent to the polymer itself, such as monomers, oligomers, or even degradation products, plasticizers, antioxidants, other additives, but also to post-processing as well. So for instance, lubricants, adhesive, cleaning agents, really any part of the manufacturing process or post-process should be under scrutiny for potential extractable leachables. I'd also like to mention when doing these E&L studies, it's important to look at the final device. So this means even after post-sterilization, for instance. I know that gamma radiation can be pretty harsh and can actually change the extractable leachable profile of your final device. So it's important to look at that final device when conducting these extractable and leachable studies. So what is the difference between an extractable and a leachable? An extractable is usually considered something that could potentially migrate from our product, whereas a leachable is the things that actually do migrate under standard use conditions. We usually like to think of a leachable as a subset of all extractables. And really the goal of these E&L studies is to find out, one, what is leaching from your product, and two, how much of that is leaching. And those are really the two main components that are needed to make a proper toxicological risk assessment of a product. So one approach to this would be to start at an extractable study. And from here, this is where we're trying to get everything that could potentially migrate out. So if we look at that previous slide, we're trying to cast a wide net to basically see everything that could potentially be a concern before moving on. And the way we do this is we use our exaggerated conditions. So whether that's an increase in temperature compared to how it will actually be used, or maybe we'll extract with a more organic solvent rather than something that would be aqueous-based. And from here, once again, we'll get our potential leachables along with semi-quantitative data, as this is really just kind of a screening platform, and we haven't zoned in on any specific impurities to quantify. So for an example, here's what a chromatogram might look like from an extraction, where I'm just showing the detector response as a function of retention time, where hopefully each one of these peaks would just represent a single compound of interest. So one thing to do here is you'd like to make some sort of toxicological risk assessment, maybe using an analytical evaluation threshold to identify which of these peaks are actually of safety concern. If you can filter this out, you can save yourself a lot of time as far as analysis goes. Let's say you would only have to identify 4 peaks rather than a couple hundred. So for instance, in this application, we'll just say that with help from a toxicological assessment, we've identified these 4 compounds of interest from a safety perspective in our extractable study. Then what we could do is then go on to a leachable study where we are following the leachability of these 4 compounds, which we can identify and therefore have standards for, and then create a validated method looking at quantitative information. I'd also like to point out that it is possible, making this assessment from your extractable studies, that there are no peaks above some sort of safety concern threshold, which would indicate that under worst case scenario conditions from that extractable study, that maybe there isn't anything of concern that would be in a leachable study. And therefore, perhaps you can use that as justification to forego the leachable study and just use that extractable data in your submission to a regulatory agency, for instance. But nonetheless, continuing on with this specific example, let's say you've identified these 4 compounds of interest that you would then like to do a leachable study on. And with a leachable study, this will be more stimulated use conditions. So let's say you'll use a phosphate buffered saline instead of ethanol, like what you would maybe use in an extractable study. And this will give us our leachable components under stimulated use, and it will also give us quantitative data with a validated method that will have various performance suitability in that method. And once you have your final leachable components, then you would want to make your final toxicological risk assessment to gauge product safety. And the way that you would want to do this is take into consideration, patient population with that example I gave earlier and the baby bottle, or as well as duration of use, whether this is a lifetime device or maybe it's just a single-use device. That will all be under consideration, as well as the route of administration. So that would be something where you're looking at an injectable or an inhalant. Those would be considered high risk versus an oral dosage form, which would be more of a low-risk application. Other design considerations that since each product really does have a different application, it will therefore need some sort of customized testing. There really isn't a one-size-fits-all study because there is, as I mentioned earlier, different application, different patient population, different dosage. All these things will be taken into account when designing this E&L study. However, when you are submitting to a regulatory agency, the biggest criteria is really justification of your study design choices. That's really what's most important. You can have sound scientific reasoning behind these choices. That's really what these agencies are looking for. It's also important to start thinking about E&L work early in product design. You don't want to go 18 months into development of this product just to find out that the materials you selected are not going to be safe from an extractable leachable standpoint. So therefore, you could throw out all that time and money just out the window without considering about E&L early in the process. It's also important to consider where the product will be marketed. For instance, in the U.S., they're very interested in looking at that whole extractable profile. The FDA really likes to see everything that could potentially be of concern migrating from a product. Whereas in the EU, they're very a little bit more focused on leachable studies themselves, as they like to see how stimulated use conditions will be used with this product. So once you have your study design, you'd like to identify some sort of testing levels of interest. So as I briefly mentioned earlier, one way to do that would be with an analytical evaluation threshold. So what that basically is that is a on-instrument detector response where you can basically draw that horizontal line through your chromatograph and decide that anything below that horizontal line, regardless of its identification, would not be of concern from a toxicological standpoint. So one way to do that would be looking at tolerable daily intake, which you would cruise the published literature for. I know that for bisphenol A, the European Food Safety Authority recently gave a limit of 4 micrograms per kilogram per day. So if we can do a calculation there, assuming patient body weight, duration of use, you could get something on instrument around 1 ppm, which might be fine and dandy for traditional mass spectrometry with a triple quadrupole detector. However, if you don't know what you have a true unknown, you might have to look into the literature guidelines, and one of them would be the M7 assessment for mutagenic impurities from the ICH. And with this, they usually recommend in a worst-case scenario is 1.5 micrograms per day as a tolerable daily intake. So if you use this 1.5 micrograms per day instead of that 4 micrograms per BPA, you're going to get an AET, or analytical evaluation threshold, at about 10 ppb, so 2 orders of magnitude lower, just because you're using worst-case scenario assumptions. And with a traditional mass spectrometry, you're really not going to be able to get down to that low of a level under traditional scanning mode. So that's where this high-resolution mass spectrometry that Waters has developed with their quadrupole time of flight has been so useful, because I've been able to get down to detection limits, below 10 ppb on instrument in scanning mode. So on top of that high-resolution mass spectrometry, which I found to be most useful for low semi-volatile organics and identification, we at PACE also like to run a suite of other testing, as well as particulate matter analysis, or looking for elemental impurities with ICP-MS, also looking at volatile organics or residual solvents with Headspace GC, and then some semi-volatile organics with direct eject GC. So here is the current Waters technology that PACE currently employs. It's a Xevo G2XS quadrupole time of flight instrument. This is a high-resolution mass spectrometer coupled with H-class UPLC, and we also use the latest unified software that is developed for this high-resolution mass spectrometry. So just going a little bit more into high-resolution MS and why it is so useful, I can show this hypothetical mass spectrum in the upper left here, showing a peak at 122 mass to charge ratio. And if we zoom in on that peak with a traditional quadrupole detector, Q, you can see that there is what's known as unit resolution, meaning you can't really differentiate compounds that are less than 1 mass unit apart. So as you can tell with this table that I've shown on the right, there are several different formulas that could actually be at 122, and it's really hard to pinpoint just one. However, with our high-resolution time-of-flight instrument, we can see that in this instance, there is actually 3 peaks at 122 instead of just one that the traditional quadrupole was not able to resolve. And this is extremely useful for identification, because as you can tell, we are getting high-resolution mass information, meaning we can get a mass with 4 or 5 decimal points. So if we take these observed masses and then compare it to this table on the right, you can see with quite high confidence the molecular formula that is associated with each of these peaks, which is very useful when you're trying to do any sort of identification, as it gives you, once again, much greater confidence in your assignment of unknowns. So here's what typical data would look like using the current setup that we have in our lab. The figure here is some negative mode high-resolution mass spectrometry data. So these 2 mass spectra show bisphenol A. So at the top, we see the low-energy MS, which is our parent ion. In this case, it will be BPA minus a proton, as well as an acetate adduct. But we also get the high-energy MS. So this is the fragmentation pattern, or when we apply collision energy to this ion, how exactly is it going to break apart? And these different fragments will then be unique to our BPA, and will also increase any confidence in identification based on that fragmentation pattern. And what's very useful for the studies that we do at PACE is that we're actually able to get this low-energy MS, so the parent ion, as well as the fragments simultaneously in one analytical run, in what Waters calls their MS to the E technology. So this really greatly reduces the amount of analysis time needed, as we only need to make 1 injection rather than repeated injections with something you might have to do on a triple quad. And with the latest unified data, it's actually very useful, because it automatically generates extracted ion current chromatograms, meaning every mass that is detected they'll automatically make an EIC. So in this instance, with BPA, you will take the intensity of this 227.10777, and it will just show the intensity of that peak as a function of time over the chromatogram. And this is extremely useful for a couple of reasons. One, you can get your retention time of any individual species, but also because since you're looking at an EIC, the background noise for this 227 ion is going to be very low in comparison to your total ion current micromatogram or tick. So what this means that on top of the technology that Waters employs, this software is able to increase the sensitivity of all detected ions, which greatly increases our signal to noise and helps us get to those really low detections. Also, once you have this low energy and high energy information along with retention time, you can create your own scientific library, which is a strategy that we employ at PACE and found to be very useful. So with this parent ion, the fragments, and the retention time, you can throw that away into a library, hold it for later, and then any time that we would see BPA in an analysis, we can match up that information directly from the library, and it will be automatically identified, which greatly reduces the amount of investigatory work needed from an analyst. So now that I've kind of talked about the approach to extractable and leachable studies at PACE, I'd like to talk about a specific case study that I recently conducted looking at 3D printed medical devices. And there has been a growing market for 3D printed devices for several reasons, probably the biggest one being that they're customizable. You can tailor them directly to the morphology of a patient, and you can create them in ways that you wouldn't be able to just traditionally off a manufacturing line. So it's very, very useful for developing exactly to the needs of the patient. It's also one integrated piece, and they're quick and inexpensive. So if you have the right equipment on staff, you can actually just do it in the same day or within a couple days rather than having to source out and having a month lead time just for one piece, whereas with this 3D printing, you can make just one piece or two pieces if you need them, rather than having to mass produce thousands of them at a factory. It's also very inexpensive, relatively speaking, as well. So there is a lot of growth in this industry, and for good reason. There are a wide variety of applications. Right now, they're being used for whether it's hearing aids or cranial implants, jaw implants, spinal cages, dental work, or really just any general implantable, and even going down to screws or hardware and the like. But really, I'd like to focus your attention to this funny-looking picture on the right. And this is actually a scanned bronchial tube of a child that had tracheal bronchomalacia. And under this condition, the child's windpipe would collapse just with regular breathing. And as you can imagine, that is a life-threatening condition that has no cure. So what doctors at the University of Michigan did is they took a CT scan of this child's bronchial tube and then were able to 3D print that out as a model shown in that photograph. And from there, also using 3D printing, they were able to make a customizable stent that could implant into the child to help keep that airway open. And upon doing this, they found that there were really no side effects, negative side effects from this surgery. And the child was off a ventilator within days and was able to lead a normal, healthy life just because of the advancements in technology that 3D printed has provided. So as you can see, there really is a growing industry into this customizable additive manufacturing. However, from my perspective at least, there is a need for some E&L testing. There have been more than 85 devices approved by the FDA that were based on this 3D printing technique. However, this approval is based on biocompatibility, which is basically a cytotoxicity test, meaning you throw this in a petri dish with some cells and will it grow? That's my crude take on biocompatibility. But however, the idea is there really isn't any chemical characterization, which is really where this field is going. And the reason why you might need some chemical characterization is because this 3D printing often involves heating plastic filament to very high temperatures. And what that could do is that could increase your potential for plasticizer leaching as well as contamination from the printer materials itself. So here I show a schematic of the printing technique that we used for this study, where you'll have a filament of polymer that you spool through a heated nozzle, and then you deposit onto a build platform to get your final printed device. And the polymers that we investigated were 4 different polymers, looking at polylactic acid as well as an FDA-approved version of that polylactic acid. And these were investigated because they're very common in implantables, and they're also a biodegradable polymer. So this is also used for food contact surfaces. So anytime you have biodegradable silverware or plastic plates, cups, things like that, that's also the same polymer. We also looked at polyethylene terephthalate, a glycol-modified version of that polymer, as well as polycarbonate, which has a variety of medical device applications. So what we did is we printed various coupons, rectangular coupons that were representative of medical devices. And we looked at 3 different colorants that were added to these devices, white, red, and yellow, to just kind of get an idea of how often these colorants would leach in an extractable leachable. And we also looked at a couple different nozzle types. So we looked at brass alloy as well as steel. Because as I described on the previous slide, you're applying a lot of heat through this nozzle to print the device. So really interested to see if there were possibly any elemental impurities that were being transferred on to our 3D printing device. And this 3D printing, when I say we, I don't actually mean myself. This was done by Dr. Zane Baird at the Indiana Biosciences Research Institute. He's the one that did all the printing and optimization of the parameters. And this is Zane right here. He says hi. So the study design for this particular application is we looked at the extraction of with solvents of various polarities, so going from high polar water all the way to something that's non-polar like hexane. And these were extracted at 50 degrees Celsius for 72 hours. And then we used a suite of analytical instrumentation that I kind of described earlier, where we're using GC headspace and direct inject for volatiles, semi-volatiles, as well as ICP for metal impurities, and then particulate matter, we followed the USP 788. And of course, our high resolution quadrupole light. As well as you can see at the bottom here, here's what the 3D printed coupons look like in this study. So jumping right into some results here, the volatile organics or residual solvents, we really didn't see anything at all, which is kind of to be expected based on the materials that were chosen. But moving on to metals, we did actually see some lead impurities based on the nozzle selection. So what we found is that the brass had actually 10 times more lead in the final 3D printed products as compared to steel. And even though these were at pretty low concentrations, it's important to note that, yes, nozzle choice did have an impact on our final extractable and leachable profiles. Also, when looking at particulate matter, here in this figure, I just show the number of particles that were observed per sample. That's for each of the different polymers that were investigated for water as well as IPA extracts. And that horizontal line in this figure is really the threshold of concern as mandated by USP 788. As you can see, just right away, in two instances, we saw particles that were well above the safety concern threshold. But really, what I'd like to focus your attention on is just these 2 FDA-approved polylactic acid polymers. So I didn't mention earlier, but in one instance, what we did is we took this FDA-approved polylactic acid and annealed it, which basically meant we put it in an oven for 1 hour at 100 degrees Celsius. And what this helped do is to help settle the polymer itself and get a better crystallization of that final device. And what we observed by putting that device through the annealing process is that it greatly reduced the number of particles observed by a 50-time decrease from levels that were far above a safety threshold of concern to something that is well below. So as you can see, this annealing process had a large impact on the particulate matter observed in an E&L profile. But I'd like to mention not only particulate matter, but as well as some of these plastic additives as well, which I'll talk about a little further. So in consideration of these plastic additives, but as well as other organics, that's where our GCEMS and LCMS techniques would come into handy. And in this figure shown on this slide, you can see just the number of compounds observed for each technique, where GCMS is blue and LCMS is in red for all of the polymers studied. And about 90% of everything that was observed was with the LCMS. So the LCMS platform was very useful, not only in identification of unknowns, but just the sheer number of things that were detected using that technique. And of the 533 unknowns that were observed total in the study, there was actually only one that overlapped between GC and LC, one that was semi-volatile and was observed using both techniques. So just to give you an idea of what a typical chromatogram might look like with our LCMS, here is the IPA extract of that FDA-approved polylactic acid. As you can see, it's quite complex, especially in the low end, around 10 minutes or so. So, going through this amount of data can be a burden for an analyst. However, we did employ a scientific library, so we did get a few matches with these peaks. So, for instance, this peak here at about 23 minutes or so, that's Ergonox 1010. That is a very common antioxidant that is used in many polymer formulations. However, despite getting a few matches with our scientific library, a lot of these peaks, especially around 10 minutes, were unidentified and required a little bit further investigation of the mass spectrometry. So here I am showing what the mass spec data look like for a peak at about 10.5 minutes. And once again, I'm showing the low energy MS with the parent ion as well as the high energy fragments. So thanks to our high resolution data, we can apply a molecular formula to these observed masses. So we can assign a C27H5409 parent ion at about 523, as well as we observed an ammonium adduct as well in the low energy spectrum. And then, as you can see in the high energy spectrum, there was this very distinct fragmentation pattern where we're seeing a loss of about 58 Daltons. And this corresponded to a C3H6 oxygen monomer. As we see, this is a very common indication that we are dealing with an oligomeric species. So what we found out is that this FDA-approved material was actually involved a copolymer as polypropylene glycol, or PPG, that was inherent to the polymer along with the traditional PLA that was observed. So what I did here is I noticed that this peak in the high energy MS at 175.1333, this was a common trimer that was observed in all these PPG oligomers. So what I did is I can take this mass, this 175, and produce an extracted ion current chromatogram, which basically is going to show the intensity of this 175 as a function of retention time. So each one of these peaks in this chromatogram are correlated with polypropylene glycol oligomer, which is very useful for identification. And I was also able to do this as well with the trimer from polylactic acid, which was at 217.0710. And as you can see, they're a very nice Gaussian distribution for both of these extracted ion currents, as well as pretty equal spacing between peaks, which is indicative of an oligomeric species, which gave us further confidence in the assignment that these were from the polymers itself. And it turns out that about 75% of all the unknowns that were observed were oligomeric in nature, which also helped greatly reduce the amount of analysis time needed for this particular study. Some other notable identifications that were made were on top of that Ergonox 1010, we also saw DEHP, which is a common plasticizer as well, in the FDA-approved polylactic acid. Now, this was a little bit interesting from my perspective because DEHP is diethylhexyl phthalate, and phthalates are kind of a hot button issue right now because they actually do have some serious toxicological concern. And I found it interesting that this DEHP was found in our FDA-approved material, but not in non-FDA-approved polylactic acid. So that's something that could possibly warrant further study. But on top of that, we also saw colorants as well. And I know a lot of doctors prefer colored materials to work with, but from an extractable and leachable standpoint, it's really just going to make things a lot trickier. So if you can avoid using colorants in your material, you're going to be better off in the long run. It'll make for a much easier analysis from a toxicological standpoint as well. And we also saw some phenyl phosphate in that polycarbonate, which are common plasticizers and also used as flame retardant. So just to talk about a little bit more about this annealing process that I mentioned earlier, this was done on just the FDA-approved polylactic acid devices. And it was put in an oven for an hour at 100 degrees Celsius to really go through this annealing process. And what we found is that not only like a 5,000% reduction in particulate matter that was observed, but also about a 50% reduction in polymer additives. So this would be all of the antioxidants and plasticizers. They also were reduced in concentration after this annealing process. So that really just goes to show that this annealing process is something that really should be considered when developing 3D printed medical devices. And also just like to note that this annealing process did not have a significant impact on the oligomer concentration. So that would be the polypropylene glycol as well as PLA itself. So on top of that, we also did a study where we looked at not only the final 3D printed devices, but we also looked at the filament itself before it was printed. We did an extractable leachable, just the filament, as well as the final 3D printed devices. And as was consistent with the previous data, only lead was observed with devices using the brass nozzle. So no lead was observed at all in the native filament, only in the 3D printed devices that use that brass nozzle, which gives you just further indication you probably shouldn't be using brass when you're 3D printing. Also, we saw an increase in particulate matter via the 3D printing process. So in this figure, this is once again number of particles per sample for each of the different polymers, and the 3D printed particle counts are in blue, whereas those for the filament are in red. And pretty much in almost every case, more particles were observed from the 3D printing process rather than just the native filaments. The exception being just regular old PLA. And I'm not exactly sure why that is, but that's something that might warrant further investigation as well. And also, in comparison to the pre-printed filament, we found a greater concentration of additives in the filament itself rather than the final printed device. So what this may indicate that the 3D printing process can cause some degradation of these additives or possibly oxidation, especially considering these are antioxidants and their main job is to become oxidized. That's what they're used for. So it's possible that that 3D printing process with that high heat induced some sort of changes to these molecules. This data is hot off the printer, so to speak, so further investigation of this data set is certainly warranted. And also, contrary to that previous finding, the DEHP was not observed in filament extracts. This concerning phthalate was only observed in the filament extracts. So that means, it was not observed in the filament extracts, only the final devices. So that means the 3D printing process may impact the leachability, so to speak, of plasticizers. If once they go through this high heat process, They're becoming more and more prevalent in extractable leachable profiles, whereas pre-printing, they would not be seen at all. So that is definitely something that could be of concern and would once again warrant further. So just to summarize this little case study, high-resolution mass spectrometry was very useful and an effective tool in E&L studies because it helped us confidently identify any of these unknowns that we're seeing, as well as provide quantitative information. And those are the two main goals of an E&L study in general. Is we want to know what is leaching from our product, as well as how much. And that tool was greatly accomplished with our high-res mass spectrometry. And the 3D printing process, it turns out, may impact the polymer additive concentration. We definitely saw evidence of that with the DEHP, as well as the antioxidants such as Ergonox 1010 that were observed. And the printing parameters themselves impacted these E&L profiles. For instance, that E&L process greatly reduced both particulate matter as well as polymer additive concentrations observed in the extracts. And then once again, nozzle choice did have an impact on metal impurities. So given this information, you would want to take the what and how much and give it to a trained toxicologist and see that these levels of these inorganics observed are likely below some sort of no observed adverse effect level from the published literature. So while I'm not a trained toxicologist, just cruising through the literature, I would kindly regard these as safe if I were put in that situation. So overall, this was a successful study, but it definitely involved a fair amount of mass spectral interpretation, and it would definitely be useful for any sort of extractable and leachable study. So if you have any questions, now would be your time to shine. Thank you very much for attending this, and thank you to Chemical and Engineering News and Waters.
Yeah, and thank you, Joe, for the presentation. Before we get into the questions, I just want to remind everyone that the slides will be available for this presentation. They'll be available for you to download them at the bottom widget after the event. So if you want to review anything, they should be there for you. We can just hop right into the questions, I think. These are all from listeners from what I see. Joel, has there been a study on leachables from metal 3D printed medical devices?
To my knowledge, there hasn't been any evaluation of, or at least published, on the metal impurities that may arise from 3D printing.
All right. If there are any follow-up questions, feel free, listeners, to submit those. Another question. How did you quantitate the oligomers?
Yeah, so this is really kind of a semi-quantitative approach, since this was a screening technique, really. So what we used is a standard. In this case, we used 2-mercaptobenzyl thiazole, mainly because it is present in both positive and negative mode, as well as UV absorbance. So we use that to do some sort of semi-quantitation on all of the unknowns that were identified.
All right. For LCMS, what kind of column and ionization method were used?
Yeah, sure. So this is a standard C18 column, and so reverse phase chromatography, and then the ionization was electrospray ionization. We find that is usually most encompassing type of ionization technique. I know there's some interest in APCI, but based on my experience, the ESI will ionize probably about 95% of the things that you're looking for.
All right. How did the extractable leachable profile from 3D printed materials compare with other processes, such as injection molding, that are done at lower temperatures?
That is actually a really good question. The focus of this study was just on the 3D printed materials themselves. So we didn't actively look at comparison to injection molding, but that's certainly a very interesting question and something that I would encourage people to look into further.
Mm-hmm. All right. You talked about drawing a threshold of concern line to reduce the number of identifications you needed to make. Can you say more about how you do that with your LC analysis and what detector you are using?
Sure. So basically, talking about the analytical evaluation threshold, you'd like to ideally look into the literature for any sort of tolerable daily intake, so or any reported from an agency such as the European Food Safety Authority, which I cited for our BPA, tolerable daily intake. Mm-hmm. And with that, you can then convert that into an on-instrument concentration based on a few assumptions about body weight or duration of use, and then as well as your extract volume. So for this study, I did look at that, just use BPA because that was the lowest TDI that was available, just kind of as a worst case scenario. But as I mentioned earlier, the best guidance to look for as far as unknowns are would be that ICH M7 guidance on mutagenic impurities, which you're going to, depending on the application, be looking at something as low as 1.5 micrograms per day. So I definitely encourage paging through that assessment for a better idea of how to form some sort of AET or safety concern threshold for your study.
All right. Did you look at antimony leaching from PET plastics? Antimony is a polymerization catalyst for this plastic and has been observed in single-use PET water bottles.
That is a good question. We did not look for that specifically with our ICP analysis, but based on that question, definitely something that we should have considered and might want to reconsider investigating some of the extracts that we retrieved.
Hey, why do we have these webinars, right? Also, how do you handle peaks you can't identify or can't find a standard for?
Yeah, that's a really good question because that can happen when you're going through large data sets like this. What you would like to do is, based on maybe the fragmentation, or you could even look at the UV trace in a pinch to get an idea of how this compound will absorb in the UV, maybe to get some sort of classification. When you're doing toxicological risk assessment, if you can get a classification or just any sort of information about your unknown of interest, that is helpful. Otherwise, we can certainly provide a chemical formula with a high degree of confidence at the very minimum with this high resolution technique, which is certainly helpful than nothing at all. So really what you would want to do is take a worst case scenario approach. So that's kind of why I mentioned that ICH guideline on mutagenic impurities earlier, is if you can just assume the worst, that this level is that 1.5 micrograms is going to be of interest, you can kind of make a safety assessment based on those assumptions.
All right, thanks. Let's see. Your example showing PLA extract looked like there were many analytes that may have been GCMS amenable. Did any additives show up in GCMS tests?
Yes, we did have overlap. Surprisingly, with just one of the compounds, and that was the DEH, or that phthalate plasticizer. I was kind of expecting to see a little bit more overlap, but it appears that a lot of those, on the low end of that LCMS chromatogram, were pretty highly oxidized, small oligomers originating from the PLA or the PPG copolymer that were used in that final device.
All right. Here's a question. What is more useful when it comes to exact quantitation? Is it triple quad or QTOF?
Good question. Useful is kind of an interesting way to put that, as they're both can be used very effectively to do quantitation. With water's latest technology, they can actually do MRM studies looking at specific ions of interest. So they have made great strides in that technology to do quantitation with this quadrupole time of flight instrument. However, I think traditionally the question is, for quantitation, you're probably going to get lower limits of detection with a triple quadrupole detector. But with that latest water's technology, they're starting to, they're comparable detection limits. But certainly in scanning mode, which is how this, extractable screening technique is done for unknowns, the quadrupole time of flight would be much better in this instance.
All right. This is actually a follow-up question from the question about how do you handle peaks you can't identify a standard for. So follow-up from the same listener. So when you have your instrument concentration threshold, how are you determining the quantification of each of the peaks? How do you account for ionization difference?
Yeah, okay. So in this AET, how we do that, into that calculation, we put like an order of magnitude safety factor, so about 10%. We'll get our AET that we calculate for on instrument, and then we'll just take 10% of that to account for any sort of changes in ionization. And we also selected our standard that we saw had the lowest ionization efficiency, so we can kind of use, once again, worst case scenario to judge which compounds are going to be analyzed the correct way from a worst case scenario standpoint.
All right. Next question. Have you investigated other 3D printing techniques besides FDM? Have you investigated, for example, DLP?
No. This was really just our first crack at it. This was our collaborator, Dr. Zainberg's expertise. So this was our initial technique that we used. But yes, there are other 3D printing techniques out there, and also do warrant investigation.
All right. Were the filaments characterized prior to printing? Was SEC-MALS used to determine starting molecular weights and distributions?
No. That is something that would, yes, be of interest as far as the identification of, what exactly the polymers weights that we're looking at. But that was not something that was done with the study. We were more focused on, the chemical characterization of extractables and leachables, so more small molecule work. However, that is a very interesting question, and I would kind of be interested in the results of that to see how this filament changed from a polymer standpoint going through that 3D printing process.
All right. What is the rationale to choose isopropyl alcohol as the solvent?
Really, this was kind of just looking at, we looked at 3 different solvents, 3 of them of varying polarity. So we went all the way up to hexane as well as water. So this was just kind of an in-between solvent that does kind of border on that more non-polar side, just to get a full range of solvents.
Did you guys consider any other intermediate solvents?
Yeah, a good solvent that has a lot of applicability is something like ethanol or 50-50 ethanol water, as it can mimic the consistency of bodily fluids quite well, and it's also something a little bit above water in the nonpolar scale that would help you get an effective extractable profile.
Right, right. All right. Did you look at the impact of changing print temperatures within the same material? Also, did the print speed itself cause any observable differences?
So yeah, the printing parameters were optimized, including speed and temperature, by my collaborator, Zane Baird. So I couldn't really speak to the specifics on that, other than he optimized them. And so we didn't do any study looking at the extractable leachable profiles from the various differences in printing parameters like that. But basically what we did is we found what works the best for printing, and then we used those devices themselves for the extraction step.
All right. And I just want to notify listeners that we won't be taking any more questions. We'll just finish the couple that we have in the box. Did you look at the impact of changing print temperatures? Oh, I'm sorry. I just asked that by mistake. Okay. Are there any public databases available for mass spectral identification of unknown extractables and leachables?
As far as public data specifically for E&L work, really not so much. I know a lot of people kind of build their own proprietary databases, which is something that we did at PACE, just targeting those compounds specifically for medical device and container closure applications. Their NIST has an LCMS library that they've kind of recently developed that has a few thousand compounds in it. However, that's still kind of a new technique, and as you know, LCMS libraries can vary greatly as opposed to a GCMS library with your EI ionization. So I would say, to answer that question, not to my knowledge that there are any E&L libraries out there, but there are certainly probably a large amount of proprietary libraries. I think Waters themselves actually has some sort of E&L library that they have developed as well.
All right. Just 1 second. Okay. How do you think recent guidelines such as BPOG and USP 661.3 will impact E&L studies?
That's a good question. So as of last year, the BPOG, which is a bioforum group that encompasses a lot of pharmaceutical companies, have tried to standardize some of this E&L work. And then the USP is a draft guideline out there as well that they released last year, also trying to standardize some of the testing. And from that BPOG, it almost seems like there's an excess of the amount of testing that is needed. A lot of the solvents, for instance, they're all aqueous-based in some respect, would not be compatible with GCMS analysis. And it calls for both ESI ionization as well as APCI, which again is mostly redundant. So there's definitely a move in the community to try to get some of these standardized testing. Whether or not it is completely needed is another argument. And as I mentioned before, there really needs to be some sort of customizability in these testing based on their different applications. So it'll be very interesting to see how this BPOG as well as this USP 661.3 get developed and in an effort to standardize some of this E&L work.
All right. That's all the time we have. Thank you again, Joel, for your fascinating presentation, and thank you, participants, for being a great audience. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you On24 for technology and production services, and thank you Waters Corp for the sponsorship that made this interactive webcast possible. For CNEN Webinars, I'm Manny Marone. Have a nice day.